This is the pre-recorded session for September 2nd, 2020. So if you go into announcements, I have, again, all the information you need for Tuesday and Wednesday in lieu of my absence. So your warm-ups, if you click here, it'll open the Google Docs. So you just go to Tuesday and Wednesday's date and you find your block and you'll do that SEL warm-up. So if you scroll down to activity on Wednesday 9/2/2020, again, I'll have the pre-recorded session link here, but it's also in the modules area. So this will just take you to your assignments in this link. And what you're doing on this day is completing steps 1 through 6. And you also have to do a discussion, and we'll get to that in a minute once we go into our actual assignment. So when you go to assignments, we're in 1.1.4, and it has been published for you. So you go into 1.1.4. So the lesson plan is, this lesson explores the collection and analysis of blood evidence collected from the scene of Anna Garcia's death. In your lab notebook, you will take notes on the composition of blood that you record from both the PLTW lesson and the teacher video presentation. You perform a presumptive test to confirm that the samples collected from the scene are actually human blood. You will watch a video showing ABO testing being performed on samples found at the scene of Anna's death. You will then design and carry out a lab that will help you interpret blood spatter patterns found in Dr. Opal's lab. You will add your conclusions for all 3 of these blood tests to the Anna Garcia case report that you are compiling. The learning goals are to describe the composition of blood in the ABO system, design an experiment to generate data, and develop and utilize a standard curve to analyze data. So part 1, what you're going to do, because again you're doing steps 1 through 6, is to access the lesson here. So again, if you're not already logged into PLTW, you might want to first, but you will click here and your lesson will open up. And it's activity 1.1.4, blood evidence. Don't worry about the distance learning support because you don't need that for this activity. It's already embedded for you. So what you're going to do is you're going to go to the section that says a drop of data and read the entire section. So when you scroll down, it says a drop of data, and you're going to read this entire section. Then you're going to go to the composition of blood, which is here, composition of blood. And you're going to title a new page in your notebook called 1.1.4 blood evidence. And you're going to respond to the blood brainstorm prompts. So that's this prompt here. Consider what you might already know about blood. Without doing any research, discuss the following questions with a partner. Use the space below the questions to jot down notes. What makes up our blood? Why is our blood such a vital importance to life? And do all living things have blood? So in actuality, what we actually did was, you're going to write this response in your notebook, but what you're going to do is you're going to post your response in this discussion. So if you click here, the discussion board will open up and you're going to respond there. So again, for this part, you're going to click this on the discussion board and you're going to respond there. And you must post your own responses, then respond to at least 2 other people's posts, giving reasons why you agree with them, explaining what their responses made you think about, or how they reminded you of something you had not mentioned in your own responses. Then you're going to complete step 1 and read the rest of the section. So you're going to read, so then you're going to do step 1, which is use the reliable internet source to research the function of the following components of blood. Plasma, red blood cells, also called erythrocytes, white blood cells, also called leukocytes, and platelets, also called thrombocytes. So you're going to do some research there using internet resources. And read the rest of the section. And you're going to read the entire rest of that section. So you're going to fill out pretty much this table. You're going to have your component, and then you're going to have the function. And then you're going to do the rest of the section. So if you fall down and scrape your knee, which component of blood would work to clot the blood from the injury? You're going to do that interactive part and everything. Then you're going to move on to the section that's called Is It Blood? You're going to read the entire section and take notes. So here it is with the Is It Blood? You're going to read this entire section, including the terminology, and take notes on it. Then you're going to get to a video. So you're going to watch the video and describe in your notebook how to safely remove contaminated gloves. Then you're going to learn about something called Kastle-Meyer. And you're not going to do any of this part because this is actually the information about if you were actually doing it. So you're going to learn about something called Casimir. So for step 4, you're going to request the Casimir blood test results for Anna Garcia's case from your teacher. So if you go to that document that I embedded for your announcements, I already uploaded the thing for it for you. So you have the positive control and the negative control, and you also have a scene that from the blood that was on the edge of the table and on the floor. So you have those analysis already embedded into your announcements. So when you open it up, it is there already for you, that picture. And you're going to respond, and then what you're going to do is respond to the reflection question in your notebook, and you're going to complete step 5. So you're skipping all of 4. You're going to respond to this reflection question. If during an experiment both your positive and negative controls produce the same results, what should you do and why? And then you're going to work on go to step 5 and describe what presumptive and confirmatory tests indicate about positive blood samples. And did the samples from the scene test positive for blood? If so, can you be certain from the blood samples in fact blood? And then based on the results, record a new hypothesis. So you're doing step 5. And then after you do that, you're going to go to step 5 of your instructions, where you're going to go to blood type and read the entire section and take notes. So you're going to go to blood typing and read the entire section and take notes. Then you're going to do this, step 6. You're going to do step 6 through 8 as written. So step 6, before you head into the lab, as in aka the presentation, use the presentation below to learn more about ABO system. So you're going to use this presentation, and you can just click here and it'll tell you more information about what you need. And you're going to learn about all of that stuff. Then you're going to do steps 9 through 10. So because we're not doing the in-class part, we're doing this part. So what you're going to do is open this PowerPoint about the virtual blood typing lab, and then you're going to complete steps 11 as written. So when you open up this PowerPoint, and you're going to have to present it in order to get the best experience. So you have to present the type. So it says a microteeter plate is made up of wells that blood samples will be dripped into. The strip is divided into 2 rows. 2 drops of blood from each individual will be tested with the antibodies for type A and type B. So columns A is type A control. Column B is type B control. AB is AB control. O is the O control. AG is Anna Garcia, so that stands for that. CS1 is crime scene 1 from blood from table. CS2 is crime scene 2, blood from the floor. EO is Elsie Opal. SG is Sam Green, Dominique Hall, Taylor Diaz, and Eric Piedmont. So you are testing, you have your control types for type A, type B, type AB, type O. Then you have the blood from the crime scene, which is CS1 and CS2, and then you have Anna Garcia, the victim's blood, and then you have all of the person of interest blood. So then you're going to keep going. So using a different pipette tip for each sample, add 50 milliliters of each control blood type sample into each of the corresponding wells. So for example, control A blood type goes into the 2 wells under the letter A. So here and here, and here. Repeat the same thing with blood. So with blood B goes here and here, and then it would just populate for all of them. And you're going to repeat for the rest of the samples. So then to use a new pipette tip to add 50 milliliters of anti-A serum into the wells in each row. The same tip can be used for all samples in row number 1. And then you're going to use anti-B serum for all the rows in well number 2. So that's all of these. You're going to use the anti-B serum for all of them, and and if you were doing this in lab, you would do that all the way down. Then you're going to wait. If this is that you were doing it in real life. So let the samples incubate undisturbed on the lab bench for 5 to 10 minutes. Then you're going to observe the wells for the presence or absence of agglutination. And agglutination has occurred if the mixture appears to be granular and thick rather than smoothie and watery. Finally, draw and record these results as instruction in step 10 of POTW 1.1.4. So you're going to draw and record these results. Use the controls in the first 4 columns of the micro plate to determine the blood types of the samples of interest in the Anna Garcia case. So if you look at blood type A, so blood type A, if you look at it, when you injected an anti-A serum, you saw that it kind of changed a little bit, but of course B didn't, because it isn't positive for A, neither is O, but AB has A and B blood types. So if you look at the crime scene, the blood that was there definitely tested positive for A. So that means a person that was blood type A, that was their blood. So if you look at like Anna has blood type A, Eric has blood type A, so does Dominique, and you can look at Samantha and see that Samantha tested positive for A and B, so her blood type is probably AB. But Elsie, Opal, and Taylor Diaz did not, so you can kind of cross them out as potential as persons of interest. And then you're going to return back to the lesson on Canvas. So we're going to hit escape, and we're going to go back to the lesson on Canvas because you did 9 through 10, and you're going to complete step 11 as written. So when you go to step 11, it says, using the information from the lab, determine your conclusions. Did you confirm the presence of blood at the scene? Why or why not? What conclusions can you make about what might have been at the scene of Anna's death? At this point of the investigation, are there any persons of interest that you would rule out? Why or why not? So remember what I was saying about like some of the ones that were blood type B or that weren't blood type A, you kind of rule them out at this point. Then for step 6, you're going to read the section and take notes, and be sure to go through all the slides embedded in the slideshow. So when you go down to blood spatter, you're going to talk about blood splatter and analysis and all that other stuff. So you have to do this entire section, and you're going to go through all of the slides. So that means you're going to have to keep clicking to get information in all of them. And then that will pretty much wrap up what you are doing on Wednesday, September 2nd, 2020. So hopefully everybody understands.